For the Bio 111 Biotechnology assignment handout, we're going to examine repeated regions of DNA as they are used to obtain a match between unknown DNA that is collected at a crime scene and compare that to the DNA of potential suspects. So in instances where DNA samples that are collected from such crime scenes are so small that they are unusable for analysis, scientists can use a technique called polymerase chain reaction, or PCR, in order to make billions of copies of the DNA sample. So what PCR does is it will essentially amplify the amount of DNA that has been collected. And this will give the investigator enough DNA samples to analyze. Only about 5% of a person's DNA actually codes for proteins, and the remaining 95% are referred to as non-coding DNA regions. And these regions vary considerably among different individuals. And these regions are what are used as the basis for identification based on DNA samples that have been collected from places like crime scenes. So non-coding regions often have repeating patterns of nucleotides, which are called short tandem repeats, or STRs, and each person has a different number of repeats with a specific within a specific region of their DNA. In addition to having the regions of their DNA that are repeated. So some people have dozens of repeats. For example, at the bottom of this page here, we see the nucleotide sequence CAT being repeated, while others may have the same repeat of CAT but fewer numbers of those repeats. So in that example, a person A has 9 CAT repeats, while person B has 5 CAT repeats at that particular DNA location. Now, a completely different individual may have a completely different sequence of repeating nucleotides. So each section of non-coding DNA that is analyzed is called a locus, or plural loci, and each individual has 2 copies for each STR locus because they inherit 1 copy from each of their parents. So that gives them a diploid chromosome and allele count for these non-coding regions, or these non-coding loci. Many different loci are used to create a DNA profile or a DNA fingerprint. So not just one locus, but many loci are used to create a DNA fingerprint. The United States uses a DNA database that's called CODIS in order to keep track of different STR loci and how many short tandem repeats each locus in a person is, or how many short tandem repeats each person has at specific STR loci. So in this example, the D3 STR locus for this individual here has a short tandem repeat that is repeated 13 times in one of their genes and 14 times in the other, which is why the graph here is showing peaks at 13 and 14. So the individual in this example is heterozygous for the STRs at the D3 locus. It just so happens that one of their STRs at the D3 locus has 13 repeats, while the other locus, or the other repeat at the D3 locus has 14 repeats. So that's why it's showing the numbers 13 and 14. If they inherit the same number of repeats from each parent, only 1 peak would be present. So, for example, if this graph showed only a peak at 13, that would mean that this person is homozygous for 13 tandem repeats at the D3 locus. So with that as our background information, we will complete an activity that matches semen samples collected from a fictitious crime scene in order to narrow down a list of possible fictitious suspects. So this is the STR profile that is obtained from 1 semen sample at this hypothetical crime scene. And what I have done is that I have used this STR profile in order to fill out the attached data sheet. And the data sheet indicates the numbers of the peaks for each STR locus. So in this example, at the first locus, which is the D8 locus, I have written the numbers 10 and 12 down, as if we return back to our profile here, we see that at the D8 locus we have peaks at 10 and 12. So that corresponds to the 10 and 12 that I have written down at that particular locus. If we move down the page here and we look at the D21 locus, I have written 25 and 26 down, because returning back to that page there at the D21 locus, we see that there are peaks for 25 and 26. As one last example here, we see that at the D7 locus, I have written 11 twice, because returning to our STR profile here, we see at the D7 locus there is only one peak at 11. So that means that this person is homozygous for 11 tandem repeats at the D7 locus. That means that both of the genes that they inherited, or both of the D7 loci that they inherited from both parents, has 11 tandem repeats. So, using the numbers of the peaks at each locus, I would ask you to continue filling out the data table using the figures in exercise 4 of the handout until you have completed a profile for the 2 semen samples, the victim and the 2 suspects. So eventually what you will see is that one of the suspects will have an identical STR profile as one of the semen samples. So this is the STR profile of semen sample 2. So in this case, I would look at the D8 locus and note that there is a peak at 10 and 12. So in my data sheet, I will record a peak at 10 and 12 for the D8 locus. At the D21 locus, I see also that 25 and 26 is present. So I also fill out a 25 and 26 there. And I would continue that until I had a complete STR profile for semen sample number 2. So you'll continue doing this for the figures that show the STR profiles for the victim, as well as for suspect 1 and suspect 2.